So let me introduce for the benefit of the audience and young audience. Many of them are there, over 100. And the event is also being streamed live on the YouTube platform. So I think many people have joined it. And there was almost no registration fee, and many young researchers are with us. So let us introduce Thomas, who is a PhD and also is a chair for the evidence-based toxicology in the Department of Environmental Health and Engineering at Johns Hopkins Bloomberg School of Public Health, Baltimore. He has a joint appointment with Whiting School of Engineering. He also holds joint appointment for molecular microbiology and immunology at the Bloomberg, at the Georgetown University, Washington, D.C., and also holds a joint appointment as a professor of pharmacology and toxicology at University of Kansas, Germany. So he's a director. He's the director of the Center for Alternatives to Animal Testing, very popularly called CAAT, of both universities. And CAT hosts the Secretariat of Evidence-Based Toxicology collaboration, the Good Red Cross practice collaboration, the Good Cell Culture practice and collaboration, the Green Toxicology collaboration, and the Industry Refinement Working Group. And he, as an investigator, he heads the Human Taxome Project, very interesting one funded by NIH Transformative Research Grant. He is chief editor of Frontier in Artificial Intelligence. He is also the former head of the European Commission for Center for Alternative Validation, very popularly, and one of the renowned and one of the rather first validation center in Spara, Italy, IBCAM, and has authored many papers, more than 500, and H-index, which makes us envious. 99, have you crossed 100, sir? H-index, something very great. So
it actually just passed 100 last week. So.
Oh. So we must congratulate you. Very rare feat. Generally, we publish about 300-400 papers publication. Then also, so it means you are well read, well cited, expert, and we are very pleased that you spare your time in maybe very chilly morning outside. So we like to hear from you on the different aspects of this alternatives. And I think you will be talking about good cell culture practices, I guess, even where also cell cultures are also a biological material. So that should also not be wasted. So like for animals should not be wasted. There should be refinement and reduction in the animal. So why we unnecessarily waste animals' cells also, which are so precious in the time of this stem cell development and also in the gene technology. So I think, Thomas, we are very keen, something very unique type of lecture it will be for the young as well as experienced toxicologists and biologists and different field people. So Thomas, I again welcome you to this forum. And I would like to just request you to kindly go ahead with your presentation if you have. And now floor is yours, sir.
Thank you very much for this nice introduction. I'm very grateful to join my Indian friends and some international ones here in this meeting. It is always a pleasure to be with you. And I have done a very nice introduction already to my talk. I want to focus today on the quality of cell culture, because that's something which I think is the only way we can really make them substitute for animal testing by being of utmost quality in what we are doing. I make my slides available for who is ever interested. I have met a QR code here. Don't worry if you're not fast enough with your camera, I will show it at the very end of my presentation again. I also have quite a few QR codes on for any papers you might be interested in following up. I deposited them in this work. Okay, before getting started, really, I would just acknowledge that we are working differently at the moment, working online. But this has also some good sides. It was very easy for me to accept this invitation without 2 days of travel. But also we see an enormous uptake of all of the international audiences. For example, these are 2 lectures. The next speaker, Lena Smirnova from my team, and for full disclosure, my wife is responsible for. And we have seen that in just 2.5 years, these 2 lectures have had almost 14,000 active learners when making them available online through Coursera. So there's good sides of the pandemic as well, not only the bad sides we're all suffering from. I would like to acknowledge that our work is sponsored by industry. You might take this as a conflict of interest. I see this as demonstrating that we are practically relevant in what we are doing, because it is strictly about alternatives to animal testing. It's not about their products. But we receive 3 times more money from philanthropic sources, which gives us a certain independence from industry. And our research is sponsored by the typical sources internationally, from Europe and from the US. And if you miss the logo of your company, or you have a rich uncle, I will always find space on this slide, which is already quite crowded. I also have some personal conflicts of interest I would like to let you know, so take everything with a grain of salt. I have involvements in biotechs and consulting other companies, both for microphysiological systems, pyrogen testing, artificial intelligence, and in being the editor for Frontiers in Artificial Intelligence. Okay, now let's get started. I think the important message of this conference used to be this microphysiological system we see over the last decade or more. We see that an enormous uptake in technologies which allow us to do something better. And this better is recreating the functionality and the architecture of some organs. These are 2 workshops we organized in 2016 and 2020. We published on them. Each have about 35 experts from different parts of the world stating the status of where these technologies are. And I highly recommend them as an overview where these technologies are and where they could be in the near future. The second of these workshops, published last year, just led to an article in Science, a perspective article we are very proud of, on 16th of September. This is a very good condensed overview about the microphysiological systems and where they are going, especially for drug development. We're going to organize a World Summit on Microphysiological Systems next year in May, June. I can be very short here because Lena Smirnova is going to give you full details on this. But this is really, I think, an important event. And one of my messages to you is I hope very much to team up more with India and other parts of Asia to make this a real World Summit. We have at the moment a very strong representation of some 45 organizations, mainly from Europe and the US. Brazil is involved, but I really would like to see also a more formal engagement, and in the future, the World Summit moving around the world really as it should be. So I want to focus today only on quality. And I will talk about aspects mainly on good cell culture practice. I will touch a bit on good in vitro reporting standards, which are in their making. And I will not really talk about validation, because I think most of these systems are still quite a bit away from validation, but we lay the ground by both doing our work well and reporting well on it, which is the key. So classical cell culture has a lot of problems. About 25% of cell lines are misidentified. They are often not the cells you think of. They're not the right organ, not the right species, not the right gender. And this 25% might sound shocking, but actually a tremendous number of cell lines being used are still HeLa cells. This was the first human cancer cell line in 1951 here in Hopkins. More than 20 tons of this have been grown so far. They have led to 110,000 plus publications. And you see, we are continuing to publish with this cell. About 300 per month are published. And there's more than 11,000 patents with this cell line. You could easily put these flasks one-on-one around the world. That's the amount of cells we have grown as scientists, as a scientific community. The problem that we are often cross-contaminating our cell lines and that HeLa overgrow other stuff, especially HeLa, is very well known. These are just a few of these publications. Here is statistics I put together at some point. You see, already in 1968, the first report came out where 18 different lines, which were supposedly not HeLa cells, were all found to be just HeLa cells. And then this continues, and even in a most recent paper out of China, 2015, where 380 cell lines were analyzed, 95 were not what the researchers thought they are, 93% of them being HeLa cells. This is the publication. This is not to single out China. This is only one report we have in hand, which is relatively recent. And it says here very clearly, 93% of the contaminants in cell lines established in laboratories of China were HeLa. So it's quite incredible how this is persistent. Discover magazine covered this in an article, discussed this whole thing, inspired by some of our articles and discussions. So this is a cartoon which shows everything was going along fine until they discovered the HeLa cell line expressed epsilon chromosomes. It's obviously cervical cancer cell lines, so female, but it's typically the opposite. You believe you have something male and you suddenly find out it's HeLa. Secondly, we are fighting with still an enormous amount of mycoplasma infection, which is not handled properly. We have all of the tools, but these are some studies. I don't want to go into any level of details, but you see here relatively high numbers in different studies over the last decades. We have to assume there's still a lot of academic labs are fighting mycoplasma infection, or not fighting them, actually are facing them. And this is leading to dramatic changes in cell culture, irrelevant results coming out. Then we have genetic instability and culture artifacts in our cell lines. A cancer cell line has about 20,000 mutations on average, dramatic differences. And this was a paper already some 5 years ago. We published that even in the same batch of cells coming from one of the leading cell culture facilities, the ATCC in the US, you can have dramatic differences in the same batch of cells between different vials. And just to show you, we worked together with Brown University here. The cells already did grow differently when we took them out. The cells reacted differently to estradiol. And this also was maintained when we swapped the cell lines between labs. So it was not the performance of our experiments. They were clearly different here in the principal component analysis of metabolites formed. So they are clearly distinct cells. They show this with respect to gene expression. So there's an enormous, this is a panel of the estrogen relevant genes, and you see a clear clustering of the Hopkins and of the Brown derived cells. Still, this is a cell line published on enormously. More than 45,000 articles have been published on this breast cancer cell line and continues to be a reference point. If you do a karyogram of these cells as they come from the cell bank, you see that some chromosomes are actually hardly to identify. Others have been multiplied. There's major transpositions. So it is really difficult to call this still a cell. It is a reshuffling and reorganization of the genome. We did a comparative genome hybridization, and we found that 10% of the genome was actually lost. 50% of the genes had less than 2 copies, and 30% had more than 2 copies. Sometimes up to 30 copies. So it is really a dramatic change to physiology, and I call this a Franken cell. We have further continued with this, and we asked ourselves, how much do then at least reflect cancer cells? In this paper from earlier this year, we show that actually they don't even reflect breast cancer. They are something completely different after some 30, 40 years in culture. We compared here MCF7 cells with 1,000 different samples, and with and without estradiol treatment. And we compared this with the breast invasive ductal carcinoma type of tissue they were derived from. And essentially, the message is very simple. They have nothing to do with the cancer cell. So they're not a reference point for this either. And this is not singly true for the MCF7 cells. So nobody has ever shown before that the same vials from one batch of cells is affected. And most of our work in the end was done with non-passaged cells, the genome hybridization, for example, showing difference in six major chromosomes. HeLa genome has been described here in this. This is an extract from a coverage in Nature. And what you can see here, very similar, lots of different copies, rearrangements. So these cells are simply not representing physiology. This is another example which I found shocking. These are commercial cells, and supposedly they have been transfected entering a single P450 enzyme, gene from humans into this cell line. And you can see how 3 different of these products by the company are completely different to the parental cell. So you're interpreting your results as I have an additional P450 enzyme in my cell line, and otherwise it's the parent cell line. In fact, due to cloning and subcloning, you have as a consequence dramatic genetic changes between the different cell lines, and you should expect dramatic differences which are not P450 related. Another problem we too often forget is antibiotic use. This is only one of many papers, but it shows that the addition of regular concentrations of antibiotics as we use in cell culture here leads to about 5,000 different genes, differentially expressed genes in this cell line. And this is just one example. There's tons of papers which are showing how the growth behavior differentiation of cells changes with these antibiotics commonly used. So we are dealing really with something which is a highly influential factor. And there's a lot of other artifacts in the way we are culturing cells. They're typically looking like pan-fried eggs sunny side up. They have hardly any cell-cell contact. Half of the cell sees plastic and half of the cell sees culture media. This has nothing to do with the tissue organization. And we typically have a single cell type here. The density of a cell culture is 0.1% of the tissue density. So we're diluting all factors, all the communication of the cells by doing so. And now to the positive side. What has been happening by moving to the microphysiological systems is we have first of all adapted to 3-dimensional cultures. And this has enormous advantages. It allows us to do many things in these cultures. It also allows us to recreate some architecture. This was an early workshop we did on 3D cultures, which again gives you a little bit of the feeling what doors are opening here with using 3-dimensional organoid type of cultures. But there's also a lot more to overcome. One of the dramatic problems in our type of cell culture is the sudden changes. Because it's the cheapest and the most convenient way of culturing cells, we typically add cell culture media and every second or third day we replace it. But this means that the environment of the cells changes within a second completely. We remove all of the waste and we replenish all of the nutrients which have gone. So this is a non-homeostatic way of culturing cells. And these non-physiological environment and these sudden changes require the cells to be highly adaptable. So this is forcing them out of differentiation into non-differentiated states where they can easily react. At the same time, there's a lot of additional effects of the traditional way of culturing in a cell, from edge effects, both on the plate and in the well. There's no demand on cell function. We are overfeeding with hard to high glucose concentrations. All our cells are diabetic in essence. There's no polarization. There's no flow on these cells. A lot of things, but many of them can be overcome by perfusion. This is what an organ-on-chip system typically is offering. It also brings constant oxygen access and everything to the cells in a continuous way. And we should be very clear. Bad cells don't get better with fancy endpoints. Whether we add omics or high content imaging, A cell culture which is not good from the beginning will never result in good results. But by using primary cells and stem cell derived systems, we have the chance of really bringing good cells in. Because this was the problem so far. What human cells could you get? You could get tumor cell lines. You could get materials from the surgeon or the pathologist, but these were patients, diseased, very small amounts, and you will never get the same material again. Only with the advent of stem cell technologies, as embryonic stem cells in 1999 and as induced pluripotent stem cells in 2006, we really now have the protocols to produce essentially every type of organ and any type of tissue. And we can also then start thinking about organ interactions. One of the shortcomings compared to animals, that we can actually create bodies on chip. And the two leading projects have actually shown 10 organs for 28 days as an interactive system. So there's a lot of possibility here. We have ourselves been involved in such an organ on chip system where we helped with biomarkers, the pharmacology and kinetics, how we can model these things. But we are also interested very much in ongoing projects in how we can actually use biomarkers in these multi-organ on chip systems. And we're focusing very much on metabolomics and microRNA expression in order to find biomarkers which can help us to analyze continuously what is happening in our cultures in a non-invasive way. This is an article which is summarizing some of these problems and transitions, so I recommend this if you want a short read, moving away from culture artifacts to the new possibilities of organotypic cell cultures. So let's come to the quality assurance of these systems. And I have a long personal history in this. I started in 1996 organizing a symposium on good cell culture practice in Berlin. We're invited on this topic for the German Cell and Tissue Culture Society, and then carried it on in 1999 at the World Conference on Alternative Methods, where we called on the scientific community to produce good cell culture practice. And I had the privilege to lead until 2002 the ECVAM task force for this. When I took over ECVAM, Sandra Kucke took this, and in 2005 we published Good Cell Culture Practice. This has been cited more than 500 times, so it's a well-read article. But it is essentially saying a few things about what you should do with cell cultures. You have to know your model and characterize it. You have to assure the quality of materials. You have to document, a very key aspect. You have to think about the hazards of your work. You have to comply with laws and regulations and ethical principles, and you need proper education. And these 6 principles are laid out and recommendations are made in this guidance. But 2005 was before induced pluripotent stem cells were even reported. So we brought in 2015 an international group of organizations together, and we started with 2 workshops, one in Europe, one in the US. And we started this work. These are the 2 workshop reports where we said, how can we apply this now to stem cell derived models, to the microphysiological systems? And good cell culture practice, this is, I think, an important message. It is related to good laboratory practice, which you will know for regulatory applications, but it is not the same. Good cell culture practice wants to be applicable to all types of cell culture, and especially also to academic research. So it's very basic in its way. So we created a task force, and these are some of the key people involved. Glenn Stacey, formerly at the UK Stem Cell Bank, David Pamies, at the time at my team, leading the GCCP program, and Sandra Kucke from the European Commission. And we published last year a draft, which was co-authored by the steering group. This was for the last year in discussion, with 350 stakeholder comments received. And we have integrated them, and the paper, the Good Cell Culture Practice 2.0 paper, is now in press and will appear in the next issue of Altex. So this is now finalized guidance, which is covering or extending GCP 2.0 to the type of systems we're discussing today. So stay tuned, please. I think it is quite important that the OECD is not only aware of this, but has been adapting a lot of the good cell culture practice work. So they published good in vitro method practices, which are very much based on good cell culture practice. They're also annexing the entire good cell culture practice 1.0 and the first workshop, which was available at the time, towards good cell culture practice 2.0. So you see, there's a very intimate relation. And I hope that now GCCP 2.0 will also feed into their and other people quality assurance programs. The second aspect, which I would like to stress, is it is not only about working well with cell cultures, but it's also about reporting properly about this. So in this paper, we summarize the state of the art of reporting standards for cell culture, and there's no single reporting standard available. Different to animal tests and many other areas of life science, we don't have a binding quality standard for reporting cell culture experiments, and certainly not for microphysiological systems. We also published in the same year a template for how to describe in vitro toxicology assays. Again, a big consensus process out of the EU Tox Risk project, which is demonstrating for a specific part of these things what should be considered. And this is now our next GCCP project. We have started to develop now reporting standards, and this is an open process. So if you're interested to join in, please contact me. We want to move forward here. The last point I would like to make before now coming to an end is this is all also very much related to our activities on evidence-based toxicology. Evidence-based toxicology is trying to translate aspects of evidence-based medicine, systematic review, risk of bias analysis, meta-analysis, to toxicology, because we think that it is about how we handle our results. This was based on an article we wrote in 2006 and the first conference we organized, both still when I was in ECVAM, the European Center for Validation of Alternative Methods of the Commission. And we then called on the international societies and the scientific community to create evidence-based toxicology as a discipline. And I can report on tremendous advances. In 2009, I got the first chair in the world for evidence-based toxicology at Hopkins. Since 2011, we are hosting the Evidence-Based Toxicology Collaboration. And this is an organization in the meantime worldwide promoting evidence-based toxicology. And in the board of trustees, we have the EPA represented, the FDA, the European Food Safety Authority, the US National Toxicology Program, and so on. And again, quality is key. An important aspect is to identify the quality of the data you're considering for your systematic review. I would like to refer you to this paper where we did a scoping review, showing you what is important to judge the quality of the evidence you're using for different types of toxicological studies. So here we analyzed, among others, 12 studies which were suggesting in vitro and in vivo study quality aspects. And an ongoing project of the EBTC here is what we call the in vitro critical appraisal tool, which is trying to give a tool at hand to assess from a paper, is this of high quality as it is reported and carried out. So it is a type of checklist approach in the end, which we are developing also in close collaboration with the US National Toxicology Program. So you see, quality is about quality in the lab, how you do it, quality, how you report it in your publications, but it's also key for the evidence integration, which we are aiming for in the end, taking regulatory decisions as the evidence-based toxicology collaboration is pursuing. And with this, I would like to close. As most of the time, I'm using a quote from John Maynard Keynes, an economist, as you might know. And he said very wisely, the difficulty lies not in the new ideas, but in escaping from the old ones. I hope I've shown you a few new ideas as to the quality control of cell cultures, and I hope that we jointly start escaping from the mistakes of the past.
Thank you. Thank you very much, Thomas, for this very insight presentation. Sometime I'm a toxicologist, we take refuge in the what is difference over the control. When your control is wrong, then what is, especially with cell lines, as you talked about the contamination of mycoplasma, sometimes endotoxins are also present there. They cause lots of inflammatory response. So, and we get confused with whether it is a toxic substance response or the cell's inherent response. So this I think one of the aspects which I also feel that generally we take refuge in comparing with the control. What is your control right, or is your control appropriate, and how conditions you match. So this happens, and I think you touched upon very important secretive research rather because it's an animal house. We can have this quality control and peer review and also oversight. But what we do and inside the CO2 incubator, generally we keep ourselves, and nobody has to oversee and have oversight over what kind of results are being generated, what kind of fetal bovine serum, fetal calf serum, what kind of media is supplemented to the cells, and cells with slight change in pH or a slight change in nutrients condition, it can be modulated to 1,000-fold, 100-fold, like that. So I think researchers should be very cautious about this new concept. At least I feel that this is also very reported. And also this some kind of very individualistic research. I think I consider cell culture kind of person's own, how much perfect the technicians and he himself is. So what kind of this playing with the cells, how to control that, including same thing happens with in silico, getting the desired results with the docking experiments and all that. Lots of, I think, quality control and good practices are desirable. You highlighted very important issues. So I like that you have spared some time for questions or answers. So if anybody has some query, so they may kindly put forward to Thomas. It's a unique opportunity to the young guys, young participants. If they have any query, they can put forward. I think I like this concept of good, and also I think it has to be one thing I just wanted to. So how it is possible? Sometimes the source of cells, you mentioned about ATCC. In India also, I think NCBS, we have National Center for Cell NCCS. So how much, what are the quality standards of the source of the cells? Do they follow any kind of good practices so that researchers are at least assured that whatever they are getting is the real one, and what are the simplest tools and techniques by that we can identify the cells? Can we do some genetic profiling of the cells? What are the tools available to the researchers to at least know whether the cells he is using is the right one, like that? Please, can you just comment, Dr. Thomas?
Yeah, I mean, you've now touched on tons of very important questions. Yeah, to start with the last one, I definitely don't want to give the impression that ATCC is not an excellent cell culture bank. Actually, I think they are, and you might have seen that I'm consulting them also. I just wanted to lay out that even at the highest level of quality assurance you can get, there's still a problem within one batch of cells. And the reason for this is, from my point of view, that we have actually heterogeneous populations. This is not one monoclonal cell type, but over time there are subpopulations forming. For example, we believe that there is something between 20 and 40% of MCF7 cells in our mix which lacks an entire chromosome. And this population differs in amount, and this is why also it is we don't get exact numbers, copy numbers. It is always a mix of these different populations, which can change probably with sedimentation of cells and depending on the exact culture conditions. There's a competition between these cells. There's a whole evolution taking place. So we have really to be concerned. There's a lot of controls introduced. Authentication of cells by the cell lines is actually pretty good. That's a very high standard nowadays. They use essentially what we use for paternity tests. So you know you have the right cell and you can check this. But it is still common practice that cell lines are passaged endlessly in the labs, that they exchange between labs. Oh, can you give me your cell line? And then the mistakes happen. And these are practices where we have to work on. And good cell culture practice is recommending them. I also liked very much when you were touching on the endotoxins, because pyrogen testing is one of my claims to fame. I've been working on this for almost 30 years now. You cannot imagine how many effects of proteins we describe in the literature are actually the endotoxin contaminations from producing them in E. coli cells or whatever. Yeah, there's so many papers around where endotoxin contamination was never excluded, and this stuff is so potent and very difficult to detect when bound to the proteins. Whenever you read this cytokine has an effect like endotoxin, you should be very careful.
Yeah. So very, very, very good topic was it for us. And if Dr. Patil, if you have questions for Dr. Thomas from other parts.
Thanks a lot, Dr. Thomas. It was really eye-opening kind of session that we had in this particular conference, because in India particularly, I'm talking about the educational institutions, we are having some shortcomings in terms of infrastructure and all. Okay, and that is where we have to be very careful. So will you give some guidelines for us academicians while taking up this kind of research, so that we can be careful when we are reporting some results from our laboratories, please?
No, I think you're completely right. I think we have to think of our papers very similar to our cell cultures. It's not about the quantity of our papers, it's about the quality of the papers. Because things which are not reproducible are simply not worth the value of the paper they're printed on. They're useless. And this is why we must assure through pressure by good peer review processes, by pressure through the funding bodies, by enabling quality assurance in the different labs, that high quality is being produced. And I think it comes with the cost. It comes with the cost of being less productive to do all of these controls. But then your publications stand. They will survive over time. They will not be shown as complete nonsense. As we have seen in so many cases, and the big discussion about the reproducibility crisis in science, I think has a lot to do with the lack of quality control and the lack of proper reporting of what we are doing.
Thank you. Thank you.
Very good. So are there any questions from the participants? If you have, otherwise I can see Lena is there, so very much enthusiastic to speak to us. And over to Dr. Patil.
Yeah, Dr. Pratiksha Alag is already there, and she's going to chair the next session. I request Dr. Pratiksha to please unmute, and we can begin with the next session. Please, Dr.
Thank you so much, Dr. Patil. I just want to make sure I'm audible.
Yeah, you're audible, ma'am. Please.
Yeah, thank you so much.
Very
good evening to one and all. Yes, good evening, Dr. Raisuddin. How are you? Yeah, so thank you so much, Dr. Patil, for having me chair the next session, which is the 6th session for the 4th annual conference of SAAEI India.